Welcome again. This is the first February meeting of the IMAG MSM Working Group on Multiscale Modeling in Viral Pandemics. It's February 3rd. We have one speaker today, so we have a little more flexibility in terms of time and discussion than usual, so I appreciate that. As always, I need to remind you that this meeting is live streamed on YouTube and is being recorded and archived and will be publicly available, so please keep that in mind. People notice the change in the masthead. Lorenzo Veschini, whose name is now spelled properly on the slide, is very generously taking over the duties of coordinator, and I know you'll be getting plenty of emails from him. Jim Sluka is still helping us in many ways, and Reinhard and I are still available to take your suggestions and criticisms. We have our usual communication channels. Please help us out, especially with regard to our YouTube channel. We have a growing archive of great talks. We would love it if people took advantage of those. And you could use our Twitter account, if you like, to help us get the word out about these meetings. Are there any announcements that we need to make? I think we have a steering committee meeting tomorrow morning, so people who are involved in the steering committee, please participate. If you think you should be involved in the steering committee and you're not receiving an invite, please contact us and we will put you on the list. There are also, I am doing a survey on shareability of multicellular modeling, spatial modeling. And so if you would like to be asked about your usage of multicellular modeling and you haven't been invited, please send me a note and I will send the survey to you. Or if you receive the survey, I ask you to, if you'd be willing to fill it out, we'd be grateful. Are there any other announcements before we get started? Reinhard, did you want to say anything about the new subgroup?
Yes, so all of you should have received an email about the new subgroup we're forming on building digital twins, medical digital twins, immune digital twins. If you're interested in participating, please contact me directly and I can tell you more about it. Thank you.
Any other announcements before we get started with the seminar and the discussion? No? So next week, we have Kevin Janes doing a second talk. He gave us a very stimulating talk early in the lifetime of this working group on stress testing his models of viral replication. Very interesting work combining experimental and computational approaches to viral replication. William is going to give us a second talk as well, so that will be great. The following week we have Giuliano scheduled to talk about some of the modeling work he's been doing. We have an open slot on February 17th if anybody would like to speak up. February 24th, we also have an open slot. So if people have ideas, want to take those second slots, that's fine. We definitely, as always, appreciate your suggestions for speakers. See a paper in the literature that just came out that you'd like to hear from the authors of, let us know. See something in the news that you would like to hear about here, let us know. Please do not be shy about suggesting names of people to speak. They could be students in your group, postdocs in your group, people down the hall, or people anywhere in the world who are doing work that you think would be relevant to us. And don't be shy about putting yourself forward, even if you've spoken in the past. If you have new work or different work you'd like to present, or you'd like to hear one of our prior speakers come back and talk about other things, that's fine too. So please do help us out. Lorenzo's been incredibly energetic in going out to find talks, and we're grateful to him for putting all the effort into building a new and energized series of seminars. Please do remember to mute your mic when you're not speaking, and we will move on then without further preface to our very loyal Hana, who's going to be talking about viral co-infections, which are, I think, pretty high on people's minds at the moment. So without further ado, I will turn it over to you.
Great. Thank you, James. Let me share screen. All right, can everyone see that okay? All right, so yeah, I'm going to talk a little bit about modeling of viral co-infections. Specifically, we're talking about respiratory tract infections, but I mean, they're fairly general models, so they could be applied elsewhere as well. The work here was primarily done as the graduate thesis project of Lubna Pinky, who is now at Eastern Virginia Medical School. All right, so there's plenty of respiratory viruses, and they circulate at different times of the year, but a lot of them do overlap. So this is kind of a schematic showing the circulation or the peak circulations of different respiratory viruses. Rhinovirus, coronavirus, these are the common coronaviruses. This was put together before COVID hit. Enterovirus, adenovirus, PIV is parainfluenza virus, RSV is respiratory syncytial virus. You might want to remember this because I use this one a fair bit. Influenza, and then MPV or HMPV, human metapneumovirus. So many of them circulate kind of in similar periods. So there is overlap in when they're circulating in the population. So the first question to ask is, just because they're circulating in the population, does that mean two of them can actually get into the same host at the same time or very close to each other? And the answer to that is actually yes. So this is just data from a couple of studies. It's kind of older now, 2014 and 2012. There have been about 20 or 30 studies now published where they've looked specifically for co-infections, primarily in patients that are hospitalized with influenza-like illnesses. But you can see that some of the proportions are fairly high. So in this study, it ranged from 36% to almost 70% of the patients tested with this particular virus had a co-infection. This study also showed a fairly high percentage. Taken all together, the 20 or 30 so studies that are now available, you get percentages anywhere from 5 to about 70% of people that are in the hospital with some sort of respiratory infection have 2 or more viruses. I've seen studies where they've detected as many as 4 in one patient. All right, so that can happen, but is it still kind of interest? Well, it's only really of interest if the co-infection will somehow clinically change the disease, right? So what are the studies saying on that? Well, those studies are also all over the place. So some studies show that co-infections are less severe than single infections. Some show that they're more severe. Some show that there's absolutely no difference. And I think the issue here is that they're just broadly lumping all co-infections together. I think people need to start looking more specifically at specific pairs of viruses, and then they might find a more consistent trend. But it's definitely not clear how all of these different viruses are interacting within the respiratory tract that could possibly lead to these different outcomes. Although many virologists and clinicians are speculating that primarily they're interacting through the immune response. And I think this assumption is mostly because if there's a change in symptoms, symptoms are largely caused by the immune response. And so that must be what changes clinical outcome. All right, so if we actually look at 2 viruses interacting, these are the next few slides will be in vitro studies that I'm showing you. There's a number of different behaviors that we see that we might want to explain. So the first is called viral interference or viral block. And here we have influenza and RSV. So influenza is the top 2 lines and RSV is the bottom 2 lines. The solid line is a single infection of each of those, and the dashed line is the co-infection. So you can see that influenza really doesn't care whether RSV is there or not. It behaves the same in either case. RSV, however, is suppressed by the presence of influenza. So that's what viral interference is, basically a suppression of one virus by the presence of another one. We can also see something called viral enhancement. And this study was a study of influenza and parainfluenza, although I'm only showing the graph of influenza here. The dashed line in this case is the single infection, whereas the solid line is the co-infection. You can see that there's been an increase in the viral load of influenza when parainfluenza is present. Okay, so that's a second behavior that we sometimes see. And the third behavior is, I've labeled it chronic infections, but it might more properly be called longer lasting infections. So this is not in vitro, this is in patients. Dark bars here are co-infections, light colors are single infections. And remember, these are all respiratory infections, so really they should only last about a week, maybe two. But if you go out here 3 weeks, you see that a lot of these co-infections are still kind of lingering on. So is there something in the process of the co-infection that's causing these longer lasting infections? And is that something we can figure out? So we have 3 things that we kind of want to try to understand about co-infections: viral interference, viral enhancement, and chronic co-infections. And this seems like a prime thing that could possibly be explained by a model. So my group, we start very simple, very basic. We have gotten into some bigger models, but with the co-infections, we started with a very straightforward assumption. We just used the basic infection model that you've probably seen before, where we have target cells, eclipse cells, which are cells that are infected but not yet producing virus, infectious cells that do produce virus, and then the virus itself. And we've basically doubled that. So we have a virus 1 and a virus 2. And the only way that the viruses are interacting is that they're trying to compete for the target cells. So both virus 1 and virus 2 are going after the same target cells. And once a cell is infected with one or the other virus, it can't be infected by the other one. So we parameterized a number of viruses, 5 different viruses actually. I'll show you the list in a second. And we basically saw 2 different behaviors with this model. So in these graphs, the solid lines are the co-infection and the dashed lines are single infections. And you can see the different viruses here, some abbreviations. On the top line, or on the top row, what we see is that when there's a co-infection, neither virus is affected all that much. So we do see a slight decrease in the viral load, maybe a slight change in the duration of the infection, but there's really not a whole lot of effect of having 2 viruses in there together. On the bottom row, we see quite clearly viral interference. All right, so we have rhinovirus here with parainfluenza virus. The rhinovirus shows absolutely no change in when it's single infection versus co-infection, but parainfluenza is suppressed. And we see the same thing with influenza and parainfluenza. Since this is a model, we can try to do a little math and understand kind of what's going on behind that, what might cause these 2 different behaviors. Well, it turns out that what determines whether you're going to get viral block or kind of the extent of viral block is actually the growth rate. So the slope of this growing portion of the viral infection curve. And if they have similar growth rates as on the right here with rhinovirus and influenza virus, you're going to see very little change in either of the viruses. If there's a very big change or very big difference in the growth rates, so rhinovirus and parainfluenza virus, rhinovirus is 13 per day and parainfluenza is just 4 per day, you're going to see viral suppression. And the idea here is really simple. It's just basic competition. If you have a faster growth rate, you're grabbing those target cells much faster than the slower growing virus. And the slower growing virus doesn't have a chance because we don't allow for target cells to be infected by another virus. We don't have target cells regenerating, so there's no other mechanism for these slow growing viruses to actually replicate. So it was kind of neat that with this little simple model, we could actually explain one of the phenomenon we were looking for, and that it actually didn't directly involve the immune response at all. It was just a simple competition model, competition idea. But unfortunately, that model does not explain viral enhancement or chronic co-infection. So we're going to start adding some features to try to capture these other behaviors. The first thing I'm going to actually look at is the chronic co-infection. Basically, to have long-lasting or chronic infections at all, you're going to need to include cell regeneration. Cell regeneration is usually neglected in respiratory infections because they're typically short compared to the average regeneration time of respiratory cells. So the infection itself is 1, maybe 2 weeks, and the regeneration rate of respiratory epithelial cells is about 30 days. And even then, once the cells are there, they don't necessarily have the right surface receptors to participate in the infection. So it could even be a little bit longer than that. But cell regeneration does provide a limitless supply of resources. And so this competition might not be as strict or as severe if we have new cells popping up that maybe that slower growing virus can grab. From now on, I'll use the case of influenza and RSV. And so what happens, you can get chronic infection when you add cell regeneration, but you actually cannot get chronic co-infection. You will only ever have one virus that persists if you have just the cell regeneration. And interestingly enough, here it's not the growth rate that matters. So influenza actually has a higher growth rate than RSV. You can kind of see the difference in the slopes. But RSV is the one that wins long-term. And what determines which one wins long-term is actually the basic reproduction number. So this is the number of secondary infections you get from a single infected cell. And you can see for influenza, it's 58, for RSV is 146. So while RSV is slower at getting the cells initially, once it has a cell, it produces more secondary infections. And in the long run, that lets it become the chronic winner, if you will. Whereas if you're looking just at acute infections, then influenza will suppress the RSV to some extent. All right, so now we've got chronic infection, but not chronic co-infection. So we need to look at some other mechanisms that might help explain some of these other behaviors. So the next assumption that we relaxed was actually the ability for the viruses to compete for the same type of cell. And this might seem like an odd thing to allow, that two completely distinct viruses infect a single cell, but there's actually now experimental evidence that this happens. So I know the pictures are not great. I tried to put a black background so you could see it a little better. But here we have on the left is the blue dye shows the cells themselves. So you can see the nucleus and kind of the outer edge of the cell. The next panel has little red dots in it that are really hard to see, but that's influenza virus. The third panel has little green dots, a little bit easier to see, but those are labeled adenoviruses. And then the final picture is everything merged together. In the final picture, you can see that there's influenza and adenoviruses both inside the cell, kind of right around the edge of it, and being produced outside the cell. So both are very important. The fact that the viruses can both get into a cell, but they can both replicate inside the cell and be produced by the cell. So this does happen, and it's been shown for a few different pairs of viruses now that they can kind of share cells together. So this is what we call superinfection. All right, so if we add superinfection, this is what our model looks like. We still have the target cells that are basically grabbing one or the other of the viruses. We allow that when a cell is in the eclipse phase for one virus, the other virus can possibly get in there and we can get the superinfected eclipse cells. In all likelihood, this is not just limited to the eclipse phase, but we're just kind of trying to keep it simple for tractability. And then you have singly infected cells, superinfected cells that are producing both viruses, and that shows up in your virus equation. Now, we do initially assume that the probability of infecting the superinfected cells is the same as just regular infecting a non-infected a regular target cell. We also initially assume that the production rates are the same, but we do allow for all of these variables to change eventually. So if you assume that the superinfected cells are actually no different than the monoinfected cells, so all of those parameters stay the same, and again, this is influenza and RSV, you actually get the same results as for just the direct competition model, which is fairly reassuring, right? RSV is slightly suppressed by influenza, and then you get acute infections. if we allow some of those parameters to change, specifically if we allow production to be changed in superinfected cells, we can see some different behaviors. So if we allow production to be enhanced, and I really have no idea what the mechanism might be here, possibly an immune suppression type mechanism, but let's just say for some reason superinfected cells are super producers, you can get viral enhancement. So here the solid lines are the co-infection again, and the dashed lines are single infection. So if you have increased production in your superinfected cells, you can see viral enhancement of one or either of the viruses. What's most interesting is when you add regeneration back into this model. If you have superinfection and regeneration, you can actually get the chronic co-infections. So again, this is influenza and RSV. We haven't changed the parameters at all. We've just added cell regeneration back into the model. And you now see that both influenza and RSV are long-lasting infections. And you have to have the superinfected cells because these are the ones that are actually maintaining the chronic co-infection. So what's happening is that influenza, being the faster growing virus, is grabbing all those target cells, and the superinfection is now allowing RSV to come in there during that eclipse phase and have a chance to infect some cells of its own. And so all of the chronic co-infection is really maintained by the superinfected cells, which are purple here. You can see that singly infected cells basically go down to 0 during the chronic phase. Now, you might think that the chronic co-infections are kind of a rare thing, but we varied the parameters in the model. So we varied the 2 infection rates of the superinfected cells and the production rates. And here, this dark red region in both the graphs is the range where you get chronic co-infections. So if you can have the superinfection and your regeneration rate is fast enough to actually produce cells that while the infection is occurring, chronic co-infection, lengthening of the infection might not be a very unusual phenomenon in these infections. So that was all work that was done before the coronavirus pandemic. When the coronavirus pandemic started, there were a few early papers back in 2020. I think these both came out in like April, May of 2020. And they started asking, where are the co-infections for coronavirus? These were kind of based on prevalence studies. So if you had influenza and coronavirus circulating at the same time, which they sort of were in Europe in the very early phase, they expected a certain number of co-infections and they just weren't seeing them. So we thought, oh, this is something that our model could potentially answer why that might be happening. The only issue was trying to find parameters for coronavirus, which at that time there wasn't a whole lot of data out there anywhere, and I actually still have not seen very nice in vitro data for coronavirus. But we grabbed some data from a patient infection, which, as you know, in patients, they usually come to the hospital and that's when you start taking data. So you don't have very good information about the growth phase, which is pretty key in our simple model. But this is the best we had. So we got some parameters and we threw that into the basic competition model with the other viruses that we already had parameters for. So the light blue line is the coronavirus. And you can see that coronavirus is basically suppressed by all of these, even para-influenza virus, which was kind of the wimpy one before. And if you calculate the growth rate of coronavirus, it's actually 1.8. It's much, much lower than any of the other viruses. So you might be kind of skeptical, as am I, because that was a single patient. The data is not great. So is there some reason to think that this might be generally true? Well, if you think that the growth rate is sort of a reflection of the incubation period of these viruses, like the time that it takes to start showing symptoms, that might be kind of a macroscopic way to measure what the growth rate is. So we looked at the incubation periods of these different viruses. And for that original coronavirus back in 2020, the incubation period was estimated at 5 to 7 days, which is much longer than any of the other incubation periods. So there is some reason to think that coronavirus might have a fairly slow growth rate as compared to these other viruses. I think that's no longer true. The Omicron variant seems to have an incubation period of 2 to 3 days is what they're saying. So it'd be kind of interesting to get some data and rerun everything to see if we are not going to see as many, or we'll see more co-infections now. But in summary, we were using just very simple models. We actually don't have an explicit immune response in there anywhere. So none of these interactions are explicitly immune response driven, but we can explain viral block as simply a competition for resources. The faster growing viruses will infect cells first, and the slower growing viruses just don't have a chance. We can explain viral enhancement if there's some sort of a stimulation of viral production in these superinfected cells. And we can explain chronic co-infection if we have superinfection and cell regeneration happening. The superinfection helps maintain these chronic co-infections by giving the slower virus a second chance to catch those target cells. I'd like to thank you all for your attention. This is the remainder of my group, some of whom are working on some more of these co-infection projects. You can see I have a little army of high school students who are, so there's people out there who are really still very interested or coming up who are really interested in this virus stuff. I am looking for a new graduate student for fall 2022. So if anybody is interested, TCU has a small graduate program, but I think we do very well with our graduates. We have 100% employment. They are fully covered funding wise for the time they're here. So it might be a nice little niche program for someone who's interested. And we're also accepting applications right now for our summer REU program. So if anybody's interested, take a look at our website and sign up for those. And I'll take questions now.
Okay, thank you very much. That was great. Please feel free to speak up, unmute, and ask away. Jeremy?
Hi, Hana. Thank you for the presentation. It was really great. One thing that also could be interesting in the framework of these co-infections is the impact of antiviral treatment. Because I suspect that with many of these co-infections involving RNA viruses, you might be in a situation where actually, if you think about co-infection in presence of SARS-CoV-2 or influenza, people would get treated for their main severe infection, and that would create a situation in which you have potentially an antiviral drug that also has an activity against the supposedly mild virus, but that would exert some sort of low selective pressure on those viruses. And I am wondering if there is not a risk here of increasing mutants and potentially resistant virus from, let's say, RSV or other supposedly mild viruses. So I was wondering if you had any opinion or comment on that.
Yeah, so there's a number of factors here that you can consider when treatment. So when we published the coronavirus being suppressed by, influenza, we actually got a call from a reporter and they're saying, are you saying people should get the flu so that they don't get coronavirus? And I was like, no, nobody should get the flu willingly. So some people are looking at using kind of milder infections to try to suppress as a treatment for more severe infections. There's also the possibility, not everybody is tested for all of these viruses. So you can treat the more severe virus and not realize that they also have something that might be a little less severe that's being suppressed. And now that you've treated the main one, that one can come back. And that might cause problems for the patients as well. But yes, there's also the issue of antivirals having multiple action against many of these viruses and unequal action, and possibly then allowing for mutations or drug-resistant mutants to occur when you're treating. So yeah, I think this is still a very new field and people don't quite know what to make of it. And, it might be very hard to do very general things. People might have to start looking specifically at different virus combinations and what you might want to do in terms of treatment specifically for, say influenza and RSV or flurona, as everybody's calling it.
Fred?
Yeah, thanks. That was a really interesting talk. So I got two questions. One is, it looks like you focused on co-infections that happened simultaneously, that both viruses showed up at the same time. Do you see sort of quirky effects if they happen at different times or initiated at different times?
Yeah, so we played with the different times. The other assumption we had is that you had the same initial inoculum.
Mhm.
Both of these things are clearly not true, or highly unlikely to be true. So for that initial competition model, we did play a lot with changing the relative initial inoculum and the time at which infections happen. So if your slower growing virus starts before your faster growing one, that then gets rid of that advantage that it had. And so you can get more equal outcomes, I guess, where neither one is suppressed very much. And also if you give it an extra boost by giving it a higher inoculum than the faster growing virus, you can negate that as well. And then conversely, if you switch that around, the faster growing one is there first, then that second one might not even show up at all.
Yeah, maybe this is a related question. So all these viruses use different cell receptors. So I assume they use subtly different sets of target cells. So what happens if the target cells are not perfectly overlapping?
Yeah, that's one we haven't investigated yet, at least model-wise. I'm aware that that's out there, and that was more likely to be a case where they're interacting through the immune response. So we have started looking at a model with interferon, but again, assuming that they're both going after the same target cell. So yeah, that gets complicated really fast.
Certainly does. Thanks.
Ruchira?
Yes, so interferon was just what I was about to ask about. So some viruses manage to suppress the interferon response. And on the other hand, then there's, which cells are producing the interferon, do they get infected by one, so they get taken out of commission? So what kinds of things are you looking at with the interferon, and do you have any preliminary findings or anything?
So with the interferon, there's been a number of different ways of modeling the effect of interferon. So the one we've chosen so far is to model interferon as either reducing production or reducing infection of the cells. And so we can have one or we eventually have both viruses stimulating production of interferon, and then kind of seeing what happens if there's a difference between changing infection or changing production of the viruses. We don't have it quite all sorted out yet. So that's one of the projects for the high school students, which might have been a little bit too much for her. But she's got all the data. So we're now trying to sit down and sort through it all. Yeah.
Yeah, I mean, the cool thing about including the immune system is that you then have like the cooperative effects of the viruses.
Yeah.
So, all right, looking forward to more such results. Yeah, and very nice. Very nice, nicely presented. And I see Dr. Pinsky is here too.
Yes.
Giuliano?
Yes, hello. I had great presentations. Thank you for that. I had to leave during part of it though, and maybe I missed it. Where is the model implemented? Is just a system of ODEs in MATLAB or something, or?
It's just a system of ODEs in Python. Yeah.
Okay. Thank you.
Other questions? I think that the interferon is always going to be on people's minds. Certainly with influenza, it's very good at shutting down interferon, type 1 interferon antiviral resistance. And so that's an interesting question. in the model structure that you have with two viruses, you can only get a steady state because of the model structure, I think. Maybe you could get some initial oscillations because you have replenishment of your susceptible population. But I don't think that model structure will allow for persistent oscillations. But I wondered if you did, say, a two-compartment model, where you had identical compartments with crosstalk, whether you could get a situation where compartment 1 was high virus 1, compartment 2 was high virus 2, and you got trade-off where you got beating back and forth between the two. So that might show up in a patient as dominance of virus 1 followed by dominance of the virus 2 in some kind of, in a simple model, periodic, in a more complicated one,
So
stochastic.
there's a few epidemiological studies with multiple virus strains where they show kind of a similar to what you're saying, but they have more than two viruses or two infectious diseases. But yeah, one will persist for a while and dominate, and then another one will take over. And the details of how that happens, I think, is very closely tied to the details of how the viruses are assumed to interact. And so I think the same thing will happen basically with these within host models. So depending on how you assume the immune or the interferon interaction or how it's implemented mathematically will probably really determine what kind of behaviors you see.
Other questions?
This is John. If there's no other real technical virus questions, you and I have talked before about your interest in the high school students, and you gave a lot of credit, mentioned them several times in your presentation. What can you tell us a little bit about just where you come up with these high school students that can work on the kind of models you're doing?
You don't know, they find me. I've had to turn a number of them away. So somehow word has gotten out. TCU actually has, well, the physics department specifically has, we call it the research apprentice program. But we have a program where we allow high school students to participate in research. And members of the department kind of do go out and promote it to local high schools. When the pandemic started is when I got an influx of students, and I think they were all at home learning virtually. And these are kind of the brighter kids, and they had nothing to do. And somehow word got around that they could do research remotely. This is stuff they can do on their computers at home. So yeah, they tracked me down.
So how do you work with them? That fascinates me.
Well, before the pandemic, they used to come in here and they would, we'd meet for a while. They'd work in the student office for a little while and then work at home. Now they've had to work very independently. So I'm really, really quite impressed with the ones that started during the pandemic and have continued. We meet over Zoom once a week. They've been attending my group meetings when they can, so they do interact with the grad student, and I have a medical student. So I think that keeps them interested. But yeah, they figure it out on their own. They email me if they're really stuck, and I'll email them back. But we meet once a week, and they're just really bright kids.
Well, if we can ever get back to some focus on dissemination and outreach, I think you got some of the greatest examples of what the profession and the discipline and you folks could be doing in the context of dissemination of the concepts and skill and outreach to the young people that are going to follow y'all. Absolutely fantastic. Thank you.
Thanks.
I'm going to jump in. I know there are two other questions that are in line, but I wanted to make a comment following on John's, which is, I don't know if you distribute these models, but I'm a big believer in this NSF resource called NanoHub, which lets you do one button execution of models. And if you'd like maybe to sit down with me for a couple of hours and maybe one or two of your high school students, I think we could bang out in an afternoon some one-click executable demonstrations so that you'd have a you could have I'm sure you already have them, but these would be distributable code in the Jupyter Notebook so that you could use those for in high school education, for example.
Okay, yeah, that'd be nice.
If you're interested, it's not.
Sure, we
Okay, I
can.
didn't mean to, but I wanted to follow on while John asked the question because I think it could be, if you're interested, that'd be. So Alex, I think you're next in the list.
Hey, thanks.
And if I'm not mistaken, I think we actually have like a 6-month-old in the meeting today.
I just wanted to say thanks for the lovely talk. I was curious if you've used your models to look at seasonal coronaviruses at all, and do they do similar to SARS-CoV-2?
So no, we didn't do the seasonal coronavirus. So in our initial looking for respiratory viruses, we wanted to have kind of consistent parameters, or at least as consistent as we could. So we were looking for specifically in vitro infections. They all had to be in HTBE culture so that the cell type wasn't an issue and it was the closest possible to real human respiratory tract. And so we only found, sufficient data for the 5 that we had: influenza, rhinovirus, parainfluenza, HMPV, and RSV. So we looked, but we couldn't find any, coronavirus in HTBE in vitro.
Thanks.
Reed, you've been very patient.
Oh, no problem. Just hanging up. Yeah, excellent talk. I have 2 quick questions, maybe less about your model, more about your opinion. So your theory is that these co-infections, as I understand it correctly, could actually be less severe than a mono infection.
Mhm.
And one thing I see as a gap, basically because we don't have like great antivirals for a lot of these common respiratory viruses. You alluded to some data that's out there. Do we need more sampling of influenza-like illness that are flu negative as sort of a baseline? And will that type of data help with this model development from folks that go to the doctor and say, I'm not feeling good, I'm strep negative, they're strep negative, and then would it be worthwhile to actually find out what they have at a finer granularity, or do you think there's enough data out there? This is my first question.
So I don't think that kind of testing will help with building models. And I don't know if clinically right now that's going to be of a whole lot of use, because like you said, we don't have good antivirals for many of these respiratory infections. I think as we start to develop better antivirals, it's probably going to be more useful to test everybody for kind of everything when they get there, because you might run the risk of treating something that's suppressing something worse, and then that worse thing comes out and you might not have a treatment for that. I think right now, because we don't have great treatment for any of them, clinically it might not be so useful. It just might be useful in terms of knowing how often that happens.
So the in vitro co-infection data would be more useful, I guess, in that scenario.
In terms of developing models, yeah.
Okay. And then I guess my second quick question is, there's a lot of talk about the origins of Omicron and how it came to be. And one of the theories is that it came from a chronically infected patient where it was hypermutated for a long period of time. Could also be a situation of a co-infection where you had coronavirus recombination. Is there any ways to model chronic chronic infection, or have you thought about modeling sort of co-infection of 2 SARS variants? Is that something?
Mutations in it?
Yeah, I know it's a whole level of, it's not a very well phrased question, but I guess the idea being that as we sort of go through this pandemic, it seems that the new waves correlate with things that are sort of drastically different in the coronavirus genome. Are there other thoughts on the radar about how you could maybe model chronic COVID or COVID co-infection.
Well, so I mean, that superinfection and cell regeneration model can certainly do chronic co-infection. I haven't thought of adding possible like mutations or, I haven't really played much with adding mutations into models and having new strains pop up. I know such models exist, but I haven't done it myself.
Okay, thanks.
Other questions? Jeremy again.
Yeah, it's not really a question, it's more a comment, but I think this field of co-infections is really likely to pop up in the next years, not only because of the pandemic, but also because of the technical, the new technological improvements that is now used in many hospitals where, just using multiplex, you can detect many respiratory viruses simultaneously. So, and this is done now kind of routinely in many hospitals. Just when patients have respiratory kind of infections, they just do any kind of multiplex with many viruses on there. So I think that, of course, in vitro data will bring a lot, but we should have a lot of much better clinical data in the future. So I think it's really a good field to invest for students.
Yeah, and I think, it can really have some impact because we're still not, I think they really need to start looking at specific viral pairs and tracking data for specific viral combinations instead of just co-infections generally, if they want to figure out, at least clinically, what they should be doing.
John.
I have another one of those maybe scientific naive kind of questions here, but the idea, and following up on Reed's question, that if two viruses both manage to infect the same cell, they issue their DNA instructions. They put out the instructions because the whole purpose of being there is to collect the material required and put it together to create copies. If you have two viruses putting out instructions, they've got to be competing for the material that's in a cell. It's finite. Would that not cause both of them to produce copies that are really defective? Like, and probably mostly not survivable ones. But if they did survive, they would be so mutated from either of the original. And I remember somebody asking me, when they talked about, viruses, co-infection, and it's, they thought like the viruses had sex or something, and so their offspring. And it's like, no, that's not how it works. And I kind of laughed naively. But now that I think about it, if you've got those two viruses inside a cell, both just putting out their instructions, doing what they do, but they have to compete for material, then the result is going to be something weird.
So, yeah, I probably don't know enough about the detail. Being a physicist, not a biologist, I probably don't know enough about the details of replication. From the experiments that the manuscripts I've read about experiments where they have, these fluorescently labeled particles. They do seem to not crosstalk a whole lot. I'm not saying it can't happen, but they seem to be able to package each virus separately. Whether there's more defective particles there than before, I don't think anyone's measured. The other thing I'm really interested in is measuring the production out of these super infected cells. Because that was one thing when we put the model together, we were sort of hoping somebody might have some idea of whether, my guess is that production will be lower in those, just because, as you were saying, there's a finite amount of materials and now it's shared between two viruses. So production should be suppressed, but is, maybe one of the viruses is better at getting the material and its production is enhanced, whereas the other one is suppressed. So it'd be interesting if somebody could actually start measuring that.
Reed, is that kind of part of what you were asking?
Yeah, in a sense. I mean, I think, it's complicated and maybe beyond the I would say, but yeah, in a super infected cell, coronaviruses can recombine. And so you got two things happening, right? You're competing for resources in the cell, which we were talking about. And then you could also have a situation where you can almost think of the genomes as like alleles, and they could actually come up with a recombination event to make something that wasn't there before. So you create sort of a new virus. And so coronaviruses are known to recombine. So you sort of have two specific questions there, I suppose. I don't know if that answers the question, but yeah, those things are possible. So yeah,
Well, so I know-
ways to model that. I don't know what the answer is right now. We're just throwing ideas out there.
Well, influenza also recombines. So if you have two strains of influenza, that recombination seems to happen not infrequently, maybe. But if you have like an adenovirus and an influenza virus, that I don't know. How similar does the genome have to be for recombination events to occur?
Yeah, I mean, it's got to be pretty common. So in the coronavirus, they're actually it's almost like a homologous recombination based on identical sequence. With the reassortment with flu, you're getting segments reassorting. When you get a DNA and RNA virus, it's probably not going to happen. Anyways, I think it's a really interesting, another element that you could add into this as well, potentially.
Yeah, well, if coronavirus could pick up NS1 from flu, it would be bad, right?
Yeah, we wouldn't want that.
So, okay.